Hello everyone. What we're going to talk about in this lecture is the structure of skeletal muscles and ultimately how together with the skeletons they translate or they allow for body to move in invertebrates and invertebrates. So these are the generic topics I will touch. In the first part of this recording, I will talk about first of all the structure of the skeletal muscles. What are they made of? Structure of muscle fibers, myofibrils, the sarcomeres, and ultimately how all of these translate into the contraction of skeletal muscles. Then in the second part, what I will focus on is how does this contraction together with different type of skeletons translate into ultimately movement in invertebrates and particularly in vertebrates. So first of all, what type of muscles are we talking about? If you recall from the lecture, introductory lecture on animal physiology and structure, we identified three different types of muscles. We have skeletal muscles, cardiac muscles, and smooth muscles. The one we're talking about here are skeletal muscles. Those muscles that have the main function to do basically almost all the voluntary type of movements in your body, moving your body parts around, and some involuntary movements such as the maintenance of the posture. We are not talking about the cardiac muscle and the smooth muscles. What are the properties of muscles in general, particularly skeletal muscles? Well, they're all excitable. So they can all be triggered in one way or the other. There is a way in which they can be stimulated ultimately to contract, and that's the second property that they have. By contraction, what we mean is that they can actually change the overall length, going from longer to shorter. That refers to the contraction. At the same time, they can go back to the original length. In that case, they can extend. That's the third property that they have altogether. And ultimately, because of their elastic property that they have, so they can contract and then usually they go back to the final length exactly because of their elastic properties. Now let's focus on the skeletal muscle, on their structure particular. Well, first of all, we are talking about a group of muscle which is huge in vertebrates particular. Here you have a picture of the muscles that can be found in human. Basically every single line you see here will correspond to a muscle that you have in your own body. So now vertebrates can have up to 600 plus type of skeletal muscle. We are not going to get into the anatomy also because I'm not an expert in anatomy. But now when we talk about skeletal muscle, no matter how many you have and so on, they are all responsible for voluntary type of movement. So I refer to as voluntary muscles. Okay. However, they can still, as we mentioned in the introductory lecture on animal structure and physiology, they can still do some involuntary type of movements such as shivering. You don't control shivering. If you're cold, you just shiver. And that's involuntary, but still is mediated by skeletal muscles. And now, the movement that they are allowed to do is due in interaction with the skeletal system and through connection between the muscle and the skeletal system mediated by a fibrous type of connective tissue referred to as tendons, which is different from ligaments which connect bone to bone instead. Now let's look at the structure of any skeletal muscle. So any skeletal muscle ultimately consists, so imagine you're taking a full muscle like this one and you slice it. Inside you will see hundreds of muscle fibers. Muscle fibers are very, very thin and long structure which correspond to single cell, which are the product of fusion of several cells. During embryogenesis, actually your skeletal muscle are made by several cells which then will fuse together, forming the muscle fibers. When they fuse together, so they form these multinucleated cells, which has this cylindrical type of structure, which will run actually from one tendon to the other one. So each muscle fiber will run the whole length of the whole muscle. Okay, and then they are connected by together with connective tissue. So in between the muscle fibers, you will find the connective tissue. Here you have a very rich network also of blood vessels which go through each muscle fibers. The reason being that these are live cells, they need maintenance, so they need to receive nutrients, oxygen. At the same time, they have also waste that they produce that has to be removed by these blood vessels. Now, when you look at an individual muscle fiber, so these long multi-cell, multinucleated cells, so this is the structure. Each one of these will be a muscle fiber, just to give you an idea. And these are the nuclei of the different cells that fuse together to form a single muscle fiber here. Now, notice, so these are multinucleated cells that will go from one end to the other one of the skeletal muscle, but inside they're actually made by smaller structures which are referred to as myofibrils. These are cylindrical structures which ultimately are the one responsible for the contraction of the muscles. That's also what gives it this striated appearance. We'll see in a second as an alternation of different bands caused by the overlapping part of the proteins that make up the myofibrils. These are very small structures. We're talking about structures that are 1/1000th of a millimeter wide. Now, what are myofibrils made of? They're made by different proteins that ultimately organize themselves, forming two types of filaments. Based on their thickness, they're referred as thick filaments and thin filaments, with the thick filaments being about 2.5 times thicker than thin filaments. We'll see later on, I'll repeat it. So, thick filaments are actually made by myosin molecules, where thin filaments are made by different type of proteins, actin, tropomyosin, troponin, and so on. The striation that you see within each muscle here is caused exactly by the overlap of the thick and thin filaments here, where it's present. So this will be the structure where these thick filaments are overlapping with the thin filaments, correspond to the darker, more dense area that you see in the skeletal muscle. So the darker areas here. Whereas where they do not overlap, so in between here, that's where actually the thin filament join together, you will see lighter, less dense area, which corresponds to the this region right here. Okay, so there is naming for this region here. So where you have the overlapping here, it's referred to as the A band. So these dark areas here will correspond to the A band. The lighter part here will correspond to the I bands. Now, A bands and I bands and so on. So the A bands correspond to dark bands. Again, they're where you have the thick filaments overlapping the thin filaments right here. Now you have also the H zone in between here. If you look carefully, going back here, you will see there's this tiny little lighter area here. See better probably in the next slides later on. But these will correspond to the H zone right here. The H zone is that zone where there is no overlapping within the darker area, within the A band, and it's also where you have the M line. The M line is that point where opposite thick filaments are actually joined together. Opposite in terms of functionality. So these will be pulling this way, these are pulling the opposite way during contraction, but they're joined together through the M line. Now, instead, when you look at the I band here, that's the lighter part in the striation of skeletal muscle. In the middle, you have what is called the Z line. Here is instead where opposite thin filaments, so that will produce an opposite result when it comes to contraction, are actually joined together in this. Now, the region between Z lines, so this Z line here and this Z line here, is referred to as the sarcomere, and this is the unit of contraction. So all the events we'll talk about later on that lead to the contraction of the muscle are taking place in each sarcomere. But as you can see, myofibrils are made by hundreds of sarcomeres placed side by side. So you have one sarcomere here, another one here, and then you have many, many more hundreds here on each side. And each one of them, though, is contracting the same way. I repeat it over and over, so that in the end, this will result in the shortening of the overall structure here. Now, summarizing what we talked about. I don't want to get you lost here. There's a lot of names and information here that we're talking about. So if you take any skeletal muscle and you section it, you will see that it's made by several muscle fibers. These are up to 1/10, 100 of a millimeter wide cylindrical structure that go from one end to the other one. They're nothing else than fused cells, so it's a multinucleated type of cells. Within the muscle fibers, these are actually, by the way, the fibers that get innervated within a muscle. Within each muscle fiber, then you will find the myofibrils. These are 1,000th of a millimeter wide. So cylindrical structure made by several proteins organized in thick and thin filaments. This is what is responsible for the striation you see in the muscles here. And now, these, within the myofibrils, you identify the sarcomere, this unit of contractions in skeletal muscles, which are found between two Z lines. Z lines are the points where thin filaments, opposite thin filaments, are joined together. So between two Z lines, you have a sarcomere, and here that's where you have the area of overlapping between the thin filaments and the thick filaments, corresponding to the dark area, more dense area here, interrupted by the M line here, this region here, and then which will correspond to the H zone. And that's with the M line being where the thick filaments, opposite thick filaments, are joined together. Okay. Moving on. Now let's look at the structure of each sarcomere. So this unit of contraction here. As we said before, there are two types of filaments found in there: the thick filaments and the thin filaments. When we look at the thick filaments, so these are about 2.5 times thicker than the thin filaments. They're not made by a single type of protein, myosin, which consists of different parts in reality. There's the head, the tail, and the junction part. Each thick filament, is made by about 200 to 300 myosin molecules wrapped together. So in each myosin, you can actually find a tail part, which is basically where they all join together. So it will be right here. These are all the tails put together, forming the main structure. And then you have the head sticking out from the filament here. The head is the one that is responsible for the connection with the actin filaments, the thin filaments, and is the one that is responsible ultimately, you see later on, of the contraction, the movement, the sliding of the thick filament over the thin filament during contraction. Second type of filaments you have is the thin filaments. The thin filaments are actually made by different proteins. So they're made by troponin, tropomyosin, and nabolin there. Now, the main one is actin, and tropomyosin too is very important. So they're all important for the contraction process. We'll see later on. So, but actin, basically, it's organized forming two different double helixes. So a double helix, so it has two strings of actin forming together that wrap around, forming this double helix. Now, nabolin is found in the middle of this double helix here. It helps with the structure itself. Now, troponin and tropomyosin are actually found on the outside. So you have two strings of troponin and tropomyosin, one for each of the two components of the helix here, the double helix here. And what tropomyosin does is actually it follows each of the component here, and it covers the active site in actin. The active site in actin is the one that is responsible for the binding of myosin heads. So the myosin head here will bind to actin through their active site here. But at rest, so when the muscle is not contracting, tropomyosin is actually covering this active site, and therefore there is no binding between the heads of myosin and the active site of actin there. Now, troponin here is connected, it's bound to tropomyosin and plays a key role on placing tropomyosin on top of the active site or away from it. And it does that. So when it's not bound to calcium, it actually keeps tropomyosin on top of the active site. But when troponin binds calcium, it will actually displace tropomyosin, moving it towards the groove here of the double helix, therefore exposing the active site and therefore allowing for actin binding to myosin and vice versa, and therefore the movement. Okay, we'll repeat it when we talk about the contraction. So let's move exactly there to the contraction. First of all, what does cause the contraction in a skeletal muscle? Well, it's caused through the activation of synapses between efferent neurons, part of the somatic system. You remember the nervous system? The peripheral nervous system can be divided between somatic and autonomic, with the somatic one responsible for basically all the voluntary movements. Okay, most of the voluntary movements, basically all the voluntary movements plus some involuntary movements like shiver and so on. Great. So the somatic nervous system here forms with efferent neurons coming straight from the spine. They form synapses, referred to as neuromuscular junctions, with each fiber within a muscle. You will see you have that nerves coming out, nerves part of the somatic system, come out from the spine and then contain several neurons and therefore several axons. Each axon will form one or more connection with different muscle fibers through neuromuscular junction. And here, when the signal is activated, you have that at these synapses, the neuron, the efferent neuron, release this neurotransmitter called acetylcholine. Acetylcholine, what it does, it binds to a sodium ligand-gated channel here in the plasma membrane of each muscle fiber. So these multinucleated cells containing multiple myofibrils. Repeating things helps. And then the binding of acetylcholine with the sodium ligand-gated channel causes an influx of sodium inside the cellular membrane here of each muscle fiber, and this causes an action potential, exactly like it happens in neurons, in postsynaptic neurons. So it causes the formation of an action potential here. How does this translate into contraction? Well, these action potential that now is being generated here through the binding of acetylcholine here, sodium coming here, then is transferred inside each muscle fiber through some extension of the cell membrane here, which go deep and around each single myofibril here. These extension here are referred to as T-tubules or transverse tubules. So they bring the action potential inside each muscle fibers. And when they do that, what they do, they cause ultimately the depolarization also of the sarcoplasmic reticulum, which is nothing else than the endoplasmic reticulum that in skeletal muscle has the key role to accumulate a lot of calcium. So the action potential reaches the sarcoplasmic reticulum, where you have voltage-gated calcium ion channels, similar to if you think about the potassium and sodium voltage-gated channels that we talked about when we're talking about the action potential in neurons. In this case though, the voltage-gated calcium ion channels, what they do, they open up when they sense an action potential. They open up and they release all the calcium that was stored inside the sarcoplasmic reticulum. They release it in the cytoplasm or cytosol in this case. Okay, and this is what this calcium coming out of the sarcoplasmic reticulum will cause the contraction of the sarcomere and ultimately of the whole muscle. What happens there? Well, everything happens at each sarcomere. So what I'm describing here is what's happening in each sarcomere. So this region here between Z lines, and it works in the same way, but obviously when you put together all the sarcomere, then you have the full contraction of each muscle fibers and then the overall muscle. Okay. In lay terms, what is going to happen here? You have calcium being released by the sarcoplasmic reticulum, and this triggers the walking ultimately of the thick filament over the thin filaments, so that the thick filaments will be moving here in this direction, left to right in this case, or sorry, right to left in this case, and left to right in this other case. By doing that, basically it's gonna shorter the distance between the two Z lines in each sarcomere. By shortening the distance through this walking, you achieve the overall contraction of the muscle. How is this happening? Well, I already anticipated that when we're talking about the thin filaments. The thin filaments, if you remember, so that's what we're talking about here. These thin filaments here are made by several proteins. You have actin that forms this double helix here. You have enabling here in the middle that helps stabilizing this double helix, and then you have tropomyosin that covers up the active site, the site that allows for the binding or the cross bridging between actin and myosin in the thick fiber. So when there is no calcium, the tropomyosin is on top of this active site. There is no binding. But then when calcium is released by this sarcoplasmic reticulum here, it will bind first to troponin here. When it binds to troponin, troponin changes structure, changes conformation, and what it does, it pulls tropomyosin away from the active site into the groove here. By doing that, it will expose the binding site on actin here. This is what is going to trigger ultimately the binding of the myosin 2 heads to the active site here. And what happens when this happens? Well, first thing you got to understand is that in resting condition, the myosin 2 heads here will be bound to ADP. Why? Because they have an ATP binding site here. And when they bind to ADP, what happens is that they break down ATP, but the energy that was in that ATP is transferred to this bond here, and overall to the structure of the myosin 2 head. So there is a lot of potential energy which now is present here in this bond here between the ADP and the myosin head here. So in resting condition, nothing happens. ADP is attached to the head and stays there. But when there is calcium there and now the active site of actin is exposed here, now the myosin head can bind to the active site. When it does that, it actually releases that potential energy that is the bond between ADP ultimately and the head here. And when it releases that, it changes conformation. It basically goes into an angle here. By doing that, I hope you can see, is actually pulling or pushing, if you want, the thin filament in one direction and the thick filament in the opposite direction. So by doing that, it's actually moving the thick filament compared to the thin filaments. Now, once this is done, this is not the end. Once this energy has been discharged, then the myosin head will be dissociated from the active site. Now, the active site in actin are still available, but the head here has already released that energy and it dissociates from that. Once it dissociates, it will bind another ATP molecule, and when it does that, it will again hydrolyze the ATP molecule, going back again in the active form, which has a lot of potential energy associated with that because of ADP bound to that again. When this happens, it will bind again to another actin, to the actin binding site here, and will do the same thing. So we'll go back doing this cycle. This is referred to as the cross bridge cycle, over and over until calcium is available in the cytosol. So if calcium is available, these active sites here on actin are always available. This cycle will keep repeating, and therefore these thick filaments will keep moving compared to the thin filaments, right? And this will result in the contraction of the sarcomere, and therefore the contraction of the myofibers and the contraction, the muscle fibers, and therefore the contraction of the skeletal muscle. This is, again, summarizing. It's always good to say things more than once. So what we have is that we have the stimulation of a muscle fiber, again, several muscle fibers, several multinucleated cells in skeletal muscle. Each one of them is controlled by a specific synapses here, neuromuscular junction. So at this neuromuscular junction, you have release of its acetylcholine when this obviously contraction has to be stimulated. Acetylcholine here opens sodium ligand-gated channels here that cause the generation of an action potential. This action potential through the T-tubule moves from the membrane, the outside membrane of the muscle fiber inside the muscle fiber to each individual myofibers. And the T-tubule here basically causes depolarization of the sarcoplasmic reticulum and therefore opening of calcium voltage gated channels, which then release all this calcium. So as a result, the activation of this neuromuscular junction here has caused the flooding of the cytosol with calcium. What is the effect? So at each sarcomere now you have that this calcium was going to displace tropomyosin from the active site of actin into the groove. And by doing that, it exposes the active site of myosin, which then activates the cross-bridge cycle, where you have the myosin, which was bound with ADP, it has a lot of energy there, potential energy, binds to the active site. When it does that, it releases that energy by changing conformation. So it bends, the myosin head bends, and by doing that, you achieve the movement of the thick filament compared to the thin filament. Once this bending has taken place, the energy has been released. The myosin head releases also the active site, binds to another ATP molecule, hydrolyzes it, and go back again to the high potential energy state here. And therefore, will form another bond here. So we'll go back at this point to this, from here to here, form another bond here with the active site on actin, releasing that energy again, bending again, causing further movement, further shortening of the space between the two Z line in the sarcomere, and therefore contraction of the sarcomere, contraction of the overall myofibril muscle fibers, and therefore the skeletal muscles there. Now, we talked about a single interaction there between a myosin head and the actin in the thin filament. In reality, if you look at the overall three-dimensional structure here, you will see that each thick filament is actually surrounded by several thin filaments here, and vice versa. So you have this hexagonal structure here, which then translates into multiple binding, multiple movement. Another important thing to remember here, which is not mentioned in your book, but it's important to remember, is that now you will always have different heads here, myosin heads, doing the cross-bridge cycle at different times. So they're not doing it all synchronized. It's not like somebody rowing. Okay, you know, when you're going on a competition, you're all rowing at the same time. You want to be synchronized. In this case, this is not all the heads are synchronized, and you don't want that to happen, because if they were synchronized, you would have a time where all the heads are not connected to the actin sites, and this will cause the relaxation of the sarcomere. But by having them synchronous, now you have that this movement keeps going and going and going, and therefore the shortening between the Z line takes place continuously. There is never a moment when there is no contact between the thick and thin filaments. All right, so that energy never gets released. The muscle keeps contracting until the calcium is removed from the cytoplasm. When does this take place? Well, it takes place when the action potential stops. Now you don't have any more acetylcholine being released and the neuromuscular junction. There is no more action potential reaching the sarcoplasmic reticulum. Now the calcium voltage gated channel, they close, and then you have calcium pumps here, ATPases. So they are primary active transporter here, which bind to ATP and pump the calcium back into the sarcoplasmic reticulum. So the concentration of calcium in the cytosol goes down, and therefore troponin releases the calcium, go back to the original conformation, allowing tropomyosin to go back, covering the active site in the actin molecules here. So myosin cannot cross bridge anymore. The myosin heads cannot cross bridge anymore with actin. There is no more binding there, and therefore you have that the thick and thin filaments are not in contact anymore. The energy gets released, and the thick filament, they slide back to the original length that they had before. So the distance between the Z lines within the sarcomeres go back to the original distance before the contraction had taken place. Now, how does this work? So how does this translate into, so the activation of one of the synapses translated into a contraction? Well, every time you have a synapse here releasing some acetylcholine, so one bout there, you get what is called a single twitch. So you have one stimulation happening here, an action potential, you're going to have a single twitch. So basically what happens there, you have, if you have a single stimulation, you have the action potential, contraction happening, but then it stops, and therefore you have the twitch. There's contraction, but then the muscle goes back to the original length. Now these different twitches will be independent from each other as long as this stimulation takes place at a time that allows for this to happen. But sometimes you may have repeated stimulation of the same neuromuscular junction that takes place at a higher frequency. So to a point where the second twitch takes place when the first one has not ended yet. Think about it. So you have the first twitch that causes the movement of the thick filaments over the thin filaments. Now, if you wait long enough, it stops, and the distance go back between the two Z line goes back to where it was before for each sarcomere, obviously. But then, instead, if you have increased this frequency here, you may have that the second twitch takes place before the two Z lines go back to the original position. So at that point, what you have is temporal summation. Do you remember when we were talking about the nervous system and the synapses between two neurons? We're talking about temporal summation as the summation, in that case, of the voltage, of the graded voltages, graded potential, sorry, that were taking place when you had the same synapses firing at with a certain frequency, right? So in this case, we're talking about same synapses firing more frequently, right? To a point where you have that the contraction is not undone yet when the second stimulation comes back in. So the first twitch was not done, and you already received the second one. So at this point, now you start having a stronger twitch. Now, when this takes place very, very, very fast, now you have that this summation takes place, but it doesn't go up indefinitely. It will plateau at a certain point, because obviously that's the maximum contraction you can get from that specific sarcomere. There's a maximum distance or minimum distance between the Z line and even the all muscle. You can contract it shorter than a certain distance, right? That's referred to as the tetanus. Okay, and that's an essential part. That's what you're basically generating if you're pushing. You're on a bench pushing a lot of weight there, and you want to hold it there. You're generating a tetanus there. So you're contracting this muscle to the. You're firing at a very high frequency, and causing that plateau ultimately in your muscles there. But this cannot go forever, the tetanus too, because at a certain point your muscle will start running out of energy. The cross bridging, if you remember, is dependent on ATP being around. Without ATP, the myosin heads cannot acquire that potential energy. And so at a certain point you start running out there, and that's where you start having what is called the fatigue. Now the contraction, no matter how much you try, start going down again. try to lift 200 pounds for 10 minutes. You'll tell me if you don't get to the fatigue point in your muscles there. Now, how are muscle controlled? There are differences between vertebrates and invertebrates. Okay, here we're talking mostly about vertebrates. Vertebrate muscles, skeletal muscles, are more complex. So they have several muscle fibers, okay, which then have inside them tens and hundreds of myofibrils. So the control of the contraction on each skeletal muscle takes place at the muscle fibers level. So each of these multinucleated cell, look at this, that's what we're talking. Each one of these will be a multinucleated cells with lots of myofibrils inside. So each of these muscle fiber is controlled by a single neuromuscular junction. However, you may have that different muscle fibers, they are controlled by a single muscle neuromuscular junction coming though from the same axon, therefore from the same neurons. What you can have here, so if this is your motor nerve that controls the contraction of this skeletal muscle, these will be made by several neurons, all of them with their body inside your spinal cord here. This is somatic system, so you have a single neuron there, and their axons coming out. So several axons, several neurons here. But each neuron here, when it reaches the muscle, might control only a few of these muscle fibers. So one neuron here will split, basically its axon, and control, for example, all of these muscle fibers here. Another neuron will control the green ones here. Another neurons will control the red ones. All of the fibers, muscle fibers that are controlled by single neurons are referred to as a motor unit. And what you have is that now, since the different muscle fibers are under control, different neurons, you can actually regulate how many of them get activated over time. They don't all get activated over time. You may have all the blue get activated all the time because they're under control of the same neurons, but they might not be activated together with the green sometimes, or sometimes they might be activated together with green, sometimes even with the red one. So by doing that, now you have a way in vertebrates, by having several motor units, to actually control the overall strength of activation of your muscle. For example, if you're lifting up, right now I have a pen in my hand, in the muscles involved in this movement, so we're talking about tens of muscles there, I'm not activating all the muscle fibers. All right, I'm activating some of them. There's no point in going maximum full speed when I don't need it, or full strength in this case. Now is that if I start picking up my son, who's not here right now, but he's weighing 40 pounds right now, well, at that point I would have to fire more motor units, right? So by having different motor units, I can control, or my nervous system can actually control the overall strength of activation of the contractions. And now the total force that is produced by the activation of the different number of motor units is referred to in this case as the spatial summation here. Now, there are different types of muscle fibers. Okay, this is overall a morphological and functional definition of muscle fibers. So in general, when you look at, again, higher vertebrates, at the skeletal muscle of higher vertebrates there, you will see that you might identify actually different type of muscle fibers based on their morphology and also their function. So you have what are referred to as slow muscle fibers. Then you have fast muscle fibers, which can be divided into aerobic and anaerobic. So now, it's not that some muscles have only one type or the other, but all muscles will have a combination of them. But having more of one type versus the other would determine ultimately the property of that muscle. You can make that muscle more resistant, let's say, or stronger in the long run, but less strong in the short run. So if you have a muscle that's necessary for sprinting, you would have a certain proportion, but if you have a muscle that is necessary for your posture, which is a long-term type of effort there, you would have maybe an opposite type of combination or proportion of these fibers. So let's look at this fiber. This table here is from your book, and it gives you several characteristics of this fiber. What you need really to remember here is how the difference between the slow and the fast fibers ultimately reflects how fast they can contract. So the slow fiber, as the name says, they contract slower than the fast aerobic and anaerobic fibers. The intensity too is lower in slow fiber compared to the intermediate, the fast aerobic and anaerobic fibers. However, the fatigue resistant is opposite to the speed and intensity. You have more fatigue resistant in slow fibers than in fast aerobic and anaerobic fiber. And all of these is a reflection on how fast myosin heads can actually hydrolyze ATP during the cross-bridge cycle within the myofibers, making these muscle fibers. Okay. Now, let's look at that. Well, the difference between them ultimately is on the head of the myosin 2 chains that make up the thick filaments. Okay. So now, if you look at the structure of the myosin making up the genetic structure, well, there you can identify three main parts. So you have the tail here, which make up basically the basic structure, the central core of the thick fibers, right? And then, so these are referred to as the tails over there. Then you have the heads here of myosin. Now you may have different types, and the difference between them is in the speed in which they hydrolyze ATP. So you have slow hydrolyzing myosin heads and fast hydrolyzing ones. And that's what really makes the difference between the fast and slow muscle fibers there. And then together with that, you have also these intermediate neck chains here, which are responsible for the movement of the heads compared to the rest of the tails there. Now, in slow muscle fibers, as we say before, they contract relatively slow. Okay, and this is because the myosin heads cannot hydrolyze ATP fast enough. However, opposite to fast muscle fibers, they don't fatigue rapidly. So these type of fibers here are very abundant in muscles, such as those involved in your posture, swimming, those that require a long-term effort. Like the fast aerobic fibers, they are rich, relatively rich in mitochondria, because since they don't do a fast type of contractions, they can keep up, they can perform the full cellular respiration, so they have lots of mitochondria and lots of myoglobin, which is the protein that's responsible for storing ultimately oxygen within the muscle. So, and because of that, they look darker than fast anaerobic fibers, which will be the lighter one here. Now, when it comes to fast muscle fibers, again, there are two types, aerobic and anaerobic. The fast aerobic, in terms of morphology, ultimately they look more like the slow aerobic fibers. They're fairly dark in color, fairly deep red. And the reason is that they have a lot of mitochondria and a lot of myoglobin as well. And they're some sort of intermediate type of fibers. They're not at the fast anaerobic fibers in terms of contractions, but at the same time, they're not also as long-term in terms of strength compared to the slow fibers. When it comes instead to the fast anaerobic fibers here, these are the typical one that you will find in sprinter or in the skeletal muscle of sprinters. They will be enriched in fast anaerobic fibers there. And since they are so fast, they are responsible for very fast but short-term type of contractions. They don't rely much on the full cellular respiration, so on the oxidative part. They rely more on glycolysis, and therefore they tend to have less mitochondria and less oxygen storing molecules, so less myoglobin, and therefore they tend to be lighter in their property. But again, they're responsible for fast construction, but in a very, very short period. They fatigue extremely easily. When you look instead, so right now we talk mostly about skeletal muscle in vertebrates, the structure we talked about. When we talk about invertebrates muscles, instead, they are slightly different. First of all, you can still find within these striated muscles, so the typical skeletal muscle, you can still find some smooth muscle cells there. The other big difference between invertebrate and vertebrate muscle is how they're controlled. Whereas invertebrates, as we talked about earlier on, you have several motor units. In invertebrate, they're usually controlled by one or very, very few, 2, 3 motor units. So much more. So either they fire as a whole or they don't fire. So there is no much modulation there. The other difference is in the way they are activated. Well, they're activated using different type of neurotransmitters. So usually they use glutamate in most of them. And this again causes an action potential or a graded potential. In that case, you would talk about a fast or a slow type of innervation. But the other big difference is that after contraction, the contraction ends not because of the removal of calcium, but oftentimes because of the activation of an inhibitory type of innervation, which can rely on different inhibitory type of neurotransmitter. For example, when you talk about clams, right? Clams, those very tasty type of invertebrates you find oftentimes living in sand in the oceans or in the seas, salty water. So the way they contract is very similar to the way vertebrate skeletal muscle contract. So their contraction is dependent on calcium. So you have the innervation of like one, very few motor units there that causes the intake of sodium and therefore the release of calcium and so on, and the contraction of the muscle. But then when it comes to relax, the relaxation is not due because of the immediate removal of calcium. Calcium eventually will be pumped back in, but is activated by actually by release of serotonin. Okay, so which basically stops the stimulating type of innervation there. Now, also here is an example of what you can see in flying insect, which again they are invertebrates as well. How does it work? For example, they're flying. Their flying is under control of a pair of muscles, which are referred to as asynchronous flight muscles, which produce the opposite effect compared to each other. They work in pairs. They work at extremely fast speed. They can produce about 600 beats per second, just to give you an idea. Because of that, they don't really rely in summation as our muscles do. Our muscles, to produce this type of frequent type of contractions there, and therefore frequent movement, in this case of wings, would have to keep firing so that you can get this activation of the muscle. In this case, in reality, several cycles of beating, let's say 50 beats, can be actually activated by a single firing of the neuron. And the reason why this happens is because the two antagonistic or asynchronous muscles that you have in there, they actually stimulate and inhibit each other directly. So what you have is that a single activation. So you have two types of muscle. First of all, you have the vertical muscle here, and then you have the longitudinal muscle here. What you have is that the activation of flying depends on the activation on neuromuscular junction. You have one firing here, which then would activate the vertical muscles that will start contracting. When they contract, they will push the wing up. But now, instead of relying on another activation here of the longitudinal muscle by neuromuscular junction, what you have is that the elongation, the contraction, of the vertical muscles here would directly, physically activate at a certain point the contraction of the longitudinal muscles. And these would, at the same time, when they start contracting, they will start pushing on the vertical muscle, causing them to stretch. So they contract, they push these muscles here, the vertical muscle outward, and this causes the lowering of the wings. Now, again, without firing the neuromuscular junction, you have the opposite effect. When these reach a certain contraction, at this point start stimulating again the vertical muscle to contract again, which at the same time causes the longitudinal muscle to relax. And then you go back to this position. They relax, the longitudinal muscle relax, the vertical muscle contract, the wings go back up. So the single firing causes several activation and inactivation by this pair of muscle, causing the movement of these wings up and down without needing several other activation. And then at a certain point you would have another activation at the neuromuscular junction, which starts another series of cycles there. But by doing that, not relying on constant activation there, you can get to such a high number of beats per second present there.